So it's my pleasure then to introduce our first speaker, Dr. José Morais. José is an associate professor and director for the Division of Geriatric Medicine at McGill University. And he's also a professor at the McGill Nutrition Center, Royal Victoria Hospital, and a director for the Quebec Network for Research on Aging. And he's going to talk to us about the very important topic of sarcopenia.
Thank you. I'm very pleased to be here. You're going to be future geriatricians and specialists for the care of the elderly. So let's see if the content of this lecture will be of some usefulness for your future practice. These were the objectives that were proposed for this lecture and probably in your package. Important considerations when we discuss sarcopenia is the fact that there is body composition changes as one ages, and the typical change is on body weight, for example, where after attaining a maximum around 50, not necessarily optimal, but after 50 or so that there is a slight trend to decrease weight. However, body composition has different compartments. And as presented in this pie chart here, if we compare a young woman with an elderly woman, for the same weight as specified in here, one can see that the compartments are not the same. An example is that fat percentage increases by about 40 to 50%. So it goes from 27 in a young woman to about 43 in an older woman, and this was taken by longitudinal data from the studies of Margaret Flink. Well, if for the same body weight there is a compartment that has increased, so this means that another one must have decreased. And in fact, what we know is that muscle mass, which is about 30% in a younger woman, comprises only 19% in this particular study in older women, so a decrease of about 40%. So everything is proportional here. All the loss in muscle is compensated by a gaining fat. But the fat is distributed not only at the abdominal area, and we will review that, but as well as infiltration into the soft tissues, in particular for us, the interest being the fat infiltrated in muscle. To start with, this is a cross-sectional area cut of the thigh using MRI that I did in our lab, comparing a younger woman with an elderly woman. And other than the reducing muscle volume that one can see by comparing the two images, there is also more fat presented here in this light purple color in the older woman. And there is consequences of that we will review soon. This is more classical, where there is increased deposits of fat in the visceral organs. And this at all levels, being it abdominal subcutaneous as well as intra-abdominal, that can see here in the turquoise color. Of interest in this particular cut of mid-abdomen at the level of L4 is the paravertebral muscles, who have an enormous amount of fat infiltrating them, which could contribute to their decreased balance, because these muscles are important for control of the trunk. In any case, these are typical changes that one sees with age. Now, there is consequences to this deposit of fat depots centrally distributed, because this type of fat is a fat that is much more metabolic active, that generates free fatty acids, and this free fatty acids contributes to insulin resistance. At the same time, this visceral fat also produces a lot of cytokines. For example, the IL-6 and TNF-alpha, which all contribute itself to protein catabolism generating or contributing to the sarcopenia. In fact, if one considers aging contributing in one side to decrease mass, muscle mass, and on the other side to increase fat, all mediated by two major components, which is exercise and diet, then one can see that they interfere with each other such that by having a decrease in muscle, which is the most active tissue of the body in terms of energy requirements, this will lead to decreasing basic metabolic rate or basal metabolic rate. And if one maintains the same food intake through life, but our basal metabolic rate is decreased, the consequences is less energy expenditure, which then will lead to fat accumulation, because our body is very efficient in retaining energy. As well, once we have more fat accumulation, there is more cytokine being produced, and these cytokines will contribute to reduce the muscle mass. And these cytokines are the ones we just mentioned about, especially this pro-inflammatory group of cytokines. So there's a relationship between muscle and fat throughout life. And this can be improved, this model, by exercise and adequate diet, and we will touch that too. Now, sarcopenia. Sarcopenia is a state where there is a significant decrease in muscle mass, and we'll review the criteria. But it's a complex decrease in muscle mediated by many factors. So far we have discussed activity or physical activity as well as nutrition, both in terms of total energy intake, because it is important, as well as protein intake. But other than these environmental factors, there is also an ongoing loss of motor neurons from the spinal cord. And we don't understand exactly why this happens, but it is the case. Not only there is less alpha motor neurons on the spine, as well as the motor units, which is the junction of the axon with the muscle itself. It's also decreased because there is less ramification of these motor neurons. And so when there is a neural stimuli, we cannot trigger as many muscle fiber contractions. The rest is very familiar to you, since you know that age, there is a change in the hormonal environment with decrease of anabolic stimuli, so less growth hormone, less testosterone as an example, but also increase of the catabolic stimuli through the cytokine productions we just mentioned. Once sarcopenia is installed, there is a relationship between sarcopenia and weakness, which is not a direct related one, because there's another component that one calls muscle quality, and we'll review that soon. But there is also a consideration that muscle, being the greatest pool of protein for the body, once it is decreased, it has implications. We already have discussed about the change in the basal metabolic rate. So, and the energy metabolism starts to be impaired, or if you want, there's a change in that. But there's also a consideration of less muscle contributing to insulin resistance. This is not so clear. However, if there is weaknesses, if there is metabolic changes, it might predispose to disability, morbidity, mortality. There's a lot of literature on that. I just presented a few papers. I do some work on the role of insulin resistance to muscle mass, but things can be extremely complex. We'll see what we can present on that a little later. Sarcopenia then. As one progresses through decades, we can see a significant decrease in muscle in general, And this happens even in the trained athletes, master athletes. It seems to be more pronounced after age 70, but this is an example of what's happening at the lower extremities. And the importance as with the bone is to attain a maximum or a peak mass for bone or for muscle, because as there is decrease over time, if one could be above of a certain disability threshold, this is just theoretical models, but makes sense to mention it, since we can prevent attaining the disability threshold by maintaining good muscle mass, but not only mass as well as strength, because the main reason for this mass is to develop strength and then to contribute to mobility. There is a vicious circle that, reason being that physical activity is such a strong factor on maintaining muscle and mobility functionality, that there is a theoretical model suggesting that once we start losing the muscle, we have a sensation of increased effort to produce activity. This unconsciously will push one to be less physically active because it's more demanding, and by being less active, there is contribution to decrease muscle even further, and this vicious circle is then initiated. Specifically at the muscle level, at histochemistry studies showing that at the muscle there is two types of fibers, mainly type 1 and type 2. These are type 2A and X. X we think is just a maturation into the type 2A. In any event, what we do know is that there is about 10% of fiber loss per decade of life, such by age 80, 90, we have lost about 40%. And we lose more of the fiber 2 than fiber 1. Fiber 2 are anaerobic fibers responsible to generate strength, whereas type 1 are aerobic, and they are more related to endurance. It's why we see a lot of older people that still do perform very nicely in marathon, but their strength in terms of weightlifting is not as good as one advances to the 70s, 80s decades. This is just microscopic electronic pictures showing that the type 1 are the dark ones, whereas the type 2 are more light in color. And then in an older person, you lose most of these light colored fibers. There's also other pictures showing, for example, very teeny type 2 fibers, which is a new thing. We don't see that in young people. And these very teeny type 2 fibers, we consider them as being type 2 fibers that have been denervated. So there is less of a neuronal stimuli to generate contraction. It's not showing in this picture. So as one loses muscle, there is also a decrease in strength. And this has been determined to be about 1 or 2% per year after age 50, because we attain our plateau of muscle mass by age 40 or so, 30, 40. Strength starts to decrease a little later, but the result is there's a continuous loss after age 50 or so. Now, there's another concept other than the strength alone, is the concept of muscle power, and this is even more impaired, or there is greater decrease in power than in strength alone. The power combines the strength and the speed at which one produces the movement, which is very important. It's a kinetic component in there, where if you slow down your speed, you have to generate even more effort to produce an action. So the power is much more impaired with age as is strength. Now, the so-called muscle quality is the evidence that for the same muscle volume, there is individuals who generate more strength than others. And this led to the concept of having a better muscle quality. And this is extremely important. Reason being that and especially because there is a relationship between this muscle volume and the amount of fat infiltrating the muscle. So you have a lot of obese individuals who have relatively reserved muscle volume, but the strength corrected for their volume is decreased. So they have a poor muscle quality. And studies using mainly CT scan and MRI have shown this muscle infiltration by fat, and these people show us the lowest muscle quality. Obviously, to produce an action such as mobility, one needs more than strength, more than power. We need coordination, we need balance. We also need an adequate cardiovascular system to produce all of this. So it's why when one assessing function is more complex than just strength. So I'm going to go over what is happening now after the description of the muscle fibers itself to why is it that the muscle is less performant. One factor already mentioned is the fact that this decreased motor units, and this has been shown in several studies. In this particular one, motor units at the level of the lower extremity, meaning gastrocnemius muscle, is a significant decrease of about 40% if one goes. These are all cross-sectional studies, by the way, and there is some generational differences between young and older individuals, but still it's quite convincing that indeed there is a decrease in motor units as one age. Muscle, it's in continuous repair, if you wish, especially if one is very physically active. There is tears and cells that dies and others that are repaired. And this is done to the satellite cells that are deposited at the basal membranes of the fibers. Now, they are dormant cells, but under trauma or other stimuli by hormonal exercise, for example, they can then be stimulated into becoming new muscle cells. They fuse and they create the fibers. And what we know is that these satellite cells, they seem to be less active. They require a greater activation stimuli in order to become myoblasts. And then the myoblasts fuse to make the muscle cell. So this is just a diagram showing that these satellite cells can be, depending on their expression in the surface receptors for certain proteins, FACS and MyoD, they can duplicate, either become myoblasts and then fuse to make fibers, or then they can renew themselves to remain satellite cells for future use, or then they can also be degraded through apoptosis pathway. But the satellite cells, is a lot of studies now looking exactly how to stimulate them, because this would be a way of replacing muscle loss with age. Another important component of muscle preservation is also mitochondrial function, because mitochondria is the manufacturer of the cell, as you know. And as one ages, there is a little bit more, not so that much more, but there is free radical production. But it's also the effect of the accumulation of these free radicals that over time contribute to deteriorate so many proteins in the body, and then the capacity to regenerate function starts to be impaired. At the same time, ROSs are not so bad because they are required, they are very cytotoxic, and they are required to fight against infections and to participate in the stimulation of the immune system. But so there is evidences that there is mitochondrial changes as one ages, and this is mainly regulated through PGC1 alpha, and then there is a more complex intracellular regulation through SIRT1 that has both a stimulatory effect on PGC1 alpha, leading to mitochondrial biogenesis, as well as stopping mitochondrial degradation, if you wish. But consider that mitochondrial function is important for muscle and for everything else in general. Now, the effect of exercise on mitochondria, it has been shown, at least in rat models, that if you compare the level of the soleus muscle, trained versus an untrained rat, you can see that they have much more mitochondrial mass, if you wish, in their muscle. And the interesting thing here is to see the link between exercise and branching amino acids. It's one class of amino acids. We call them essential amino acids because our body cannot produce them. We must obtain them from food. But the combination of exercise with enough branching of amino acid have been shown, at least in muscle of rats, to increase mitochondrial mass, which is important to generate strength and energy. Okay, this was just a couple of factors related to aging muscle and how to preserve aging. We're going to go into something a little bit more clinical by starting with the definition of sarcopenia. The term sarcopenia, sarcos means red meat, and penia is lack of. And this was proposed by Irving Rosenberg in fact in the late 80s. And then he was just encouraging people to dedicate attention and research into why is it that one loses muscle. And in his definition, it was just a question of muscle mass loss. And later on, Baumgartner have defined a way of determining sarcopenia when he used DEXA. The DEXA that one uses to measure bone density can also be used, because of the difference in density of tissue, to determine the amount of lean tissue as well as fat. And he has proposed to define muscle mass index similar to osteoporosis, which consists of determining the amount of appendicular muscle mass, so the muscle in both upper and lower extremities, divided by the height square of patients and of individuals. And if this value is less than 2 standard deviations of the mean of a reference younger population, then we can consider the person to be sarcopenic. And what we have shown is that in a population between 65 to 70 years of age, the sarcopenia has a prevalence of about 10 to 20%, 25% sometimes. Whereas when you go above 80, it can attain more than 50%. So it's a question of time, the effect of time on muscle. Now, what are the consequences? In this study, it showed that sarcopenia was associated with difficulties in IADLs, both in men and women, and also more mobility issues with the need to use a cane. These results have been replicated in other populations, but the relationship between muscle mass, sarcopenia, with mobility is not so clear. One can also use other modalities of assessing muscle, and one that is very practical is to use bioimpedance analysis, which is just an electrical device where we apply electrodes, and it's a very tiny current that goes from foot to ends, and depending on resistance and impedance, one can then calculate the amount of linked tissue as well as fat. And Jensen, that is now working at Kingston University, proposed that you can calculate a skeletal muscle index with that, which is skeletal muscle mass divided by body weight times 100 is a percentage. And they have even proposed class 1 and class 2 sarcopenia. Class 1 being if you are less than 1 standard deviation of a mean of a reference young population, whereas class 2 is 2 standard deviations, similar to osteoporosis if you have 1 or 2 standard deviations below the mean. In this study, using data from AIMS 3 data, it showed that sarcopenia had 2 to 3, 4 times greater function impairment and disability compared with non-sarcopenic individuals. And we can use other ways of defining sarcopenia. If you don't have a reference population, because there is difference in ethnicities, in regions, etc., one approach is to define quantiles and then define sarcopenia as the 2 lowest quantiles, and there is results being published with that. So this was for definition, but other terminology have been added. Have you ever heard about sarcopenic obesity? Because there is certain individuals where the decrease in muscle is so striking and the increase in fat is so obvious that we were wondering if there is an impact on physical function. So there has been a combination of these 2 body components and see if this adds further risk for functional impairment or mobility issues, etc. So, and again, obesity defined by Baumgartner here, based on percent body fat, you can determine this if you have a DEXA or if you have DIA, for example. He found that if women have less than 27% of body fat, of men, and women should be more than 38% for women, and for men more than 27% of body fat, this will consider the person to be obese, and combine that with the muscle mass index values, then it determined that first, the prevalence of sarcopenic obesity in this study was not so high. It was 2% of individuals between 50 and 60 years of age, and 10% if they were more than 80 years old. Now, there's a lot of research on sarcopenic obesity, but the percentage is not so high, because most people who are obese, they have also more muscle mass, okay? Because their extra weight that they carry contributes to maintain the muscle, because it is a resistance type of exercise. In any case, people who are sarcopenic obese in general, it was found that they have 2 to 3 times higher IVL's disability in a prospective way, and compared to sarcopenic alone. Obviously, one can very quickly go into defining who is obese based on WHO criteria for BMI of more than 30, which can facilitate the determination of who is obese or not. However, because it's not easily accessible to have these instruments to measure muscle mass, it becomes difficult in practice to determine which of our patients could be sarcopenic. But we have this clinical intuition that tells us that most of our patients are sarcopenic. Fine. Now, things became a bit more less clear, let's put it this way, when more recent definitions of sarcopenia have also included some measures of function. I'm saying that mainly because muscle is supposed to generate strength and contribute to function. And then when together with sarcopenia we start adding other components to its definition, the difference between sarcopenia and frailty is not so clear anymore. I'm saying that mainly because the European Working Group of Sarcopenia in older people have proposed that together with low muscle mass, one should also see concomitantly with that a decrease in strength or a decrease in function, strength being measured by end grip and function by either gait speed or another test, the short physical performance battery test proposed by Morais several years ago. Well, then, you know, this is one way of describing frailty, and Dr. Rockwood will come later to explain to you this and other models, including his own work. But, you know, amongst 3 of the 5 criteria used in the phenotype of frailty by Linda Fried, one have low body mass, or if you wish, weight loss or sarcopenia. One has weaknesses and slowness that are both part of the description of sarcopenia as well as of frailty, and things are not so clear. What can we say about sarcopenia and frailty? Are they exactly the same? But there is if you wish, a lot of commonalities between both. So to make things a little bit more complex, not only we start defining sarcopenic obesity, we'll start also defining dynapenic obesity. The term dynapenia stands for lack of strength, and using NAIDS 3 data, Daniel Bouchard, who is now in Manitoba, have come up with 3 stages of, in fact, 4 groups: dynapenic obesity, obese people known as dynapenic, dynapenic alone, or normal. And what she has found is that dynapenic obesity group has slower gait speed compared to obese and normal individuals, but not compared with dynapenic individuals alone. So things have become very complex, but all the efforts here is to link body compartments with function. And we try to have a step about this relationship by using data from the Morais study. This is a Quebec study, a longitudinal study, where we reach a group of representative group of older people from Quebec, from the area of Montreal and Sherbrooke, grouped into 3 age groups: 78 to 72, 73 to 77, and 78 to 82 at inception. We followed them for 5 years, and in fact, there was about 1,800 that met the criteria. But we have body composition, measurements of the nutrition status, as well as physical performance in about 1,300, and we eliminated some of the diabetic subjects, or all these diabetic subjects, because diabetes in itself has more impact in function than the rest of the population. So we have data in more than 1,000 patients for what I'm going to present to you, and we did calculate their muscle mass and physical performance. We then classify them according to being sarcopenic or being obese, and having both or none, so in 4 different groups. And these groups were compared by general linear model statistics, including covariance of age, sex, smoking, chronic diseases, and physical activity as good factors. And here are some results. I was trying to rearrange my circles to make this clear, but we'll go together through all of this. In fact, the sarcopenic individuals, they were older in age than the older and the normal subjects. And by definition, sarcopenic individuals had lower muscle mass index compared to obese. Again, by definition, obese people had more fat. BMI was different between the four groups. Of interest is time up and go that was, if you wish, faster, not faster, it was less in normal people compared to obese people, and gait speed also was higher in normal people compared with obese. And sarcopenic people also were as, their gait speed was as fast as that of the normal. In terms of strength, to start with, for balance, obese people, this is not what I want to show. Okay, so I just wanted to say, in terms of one leg stand, which is a measure of balance, obese people, they couldn't stand as for so many seconds as normal people or sarcopenic people. Whereas in terms of strength, all the sarcopenic individuals, in average, had lower strength of upper and lower extremities. So the conclusions from this this study was that sarcopenia, which is not new, contributes to lower strength in several group muscles, but has no impact on time up and go and gait speed. Whereas obesity affects function through balance problems, and this is one reason why the obese people seems to have much more functional problems than who have sarcopenic people. Unless the sarcopenia is so severe that there is also an impact in strength. We try to understand a bit more the relationship of all of these factors in terms of body composition. This is body fat and muscle mass, its implication with strength, with mobility, and physical activity. And this with independent factors such as age, sex, energy intake to represent food intake, as well as presence of chronic disease. And for that we use path analysis, which is a combination of structural equation modeling and taking account of collinearity between all of these variables. And this model I've shown to have a very good fit using these fit analysis tests. And what we learned from this model is that mobility and strength are significantly associated. The stronger you are, the more mobile you should be. Mobility has a strong impact on physical activity. One cannot initiate physical activity if there is a lot of mobility issues to start with. So we have to work on mobility to then encourage older people to be physically active. Interesting enough, fat mass as well as muscle mass were both negatively associated with mobility. How can this be? In this model, we took account of strength. So once we take account of strength, muscle mass by itself is not so important anymore. Okay? Because what counts is the strength that favors mobility. And there is some implications one can learn from a model like this, is that greater muscle mass may not be the best target to enhance mobility. And since fat mass is also negatively associated with mobility, weight loss should be desirable in this population, right? In the population in which there is excess body fat, not in our normal patient population, because weight loss contributes tremendously to also to muscle mass too. And you know that muscle mass is needed for strength. So things are very complex. Unless we have a type of exercise that can at the same time that favors fat loss, there is a gain in muscle, and this is done through a resistant type of exercise. In terms of exercise, and we'll finish with that. This is a picture of Olga Kotelko. She's a 93 years old lady who has all the world records for track and field. I had her in our laboratory in whom I did muscle biopsies, and her mitochondrial function is something to see. And I'm not reporting that. What I'm reporting is, in general, we're very sedentary. And this is data from the Association of American Association for Retired People, showing that about 50% of our older population do some type of exercise. Only about 10% will do very vigorous exercise. And exercise is the best anti-aging strategy. But exercise for older people has to be a combination of both resistance as well as endurance, combined with balanced exercise. And this is what has been proposed by the Association of Physical Medicine as well as the American Heart Association in the publication 2007. In general, any exercise, because people sometimes ask, is it any improvement if I lose a little bit? The literature tells us that a little bit is better than nothing. Obviously, if you do exercise in the gym, it will be better, if you run, etc., but anything is better than nothing. And the effect of exercise is not only physically, it's also mentally, cognitively, as reported, and also metabolically, from the standpoint of controlling adiposity and favoring insulin sensitivity. And even in the very frail older persons, this study by Binder, published already 10 years ago, is a very good example of that, in which people undergo training exercise at a much better physical performance score than the ones who just had flexibility exercise. There's also a question of protein. I will finish with that, because studies from the Health ABC cohort have shown that people who eat about 1.2 grams of protein per body weight per day, that longitudinally over 3 years, 40% less of lean tissue loss. So this is a reason why protein intake for older people should be 1.2 and not 0.8. We know that there is some amino acid resistance with aging, meaning that older people do not benefit from, I would say otherwise, older people, they require a bigger bolus of amino acids to trigger protein synthesis. In this study, when 7 grams of essential amino acids were given, older people had less muscle anabolism, whereas in this other study, where amino acids were given as 15 grams, we could not see any difference between young and elderly here, young in black, and both of them responded to amino acids similarly compared with their post-absorptive state. And there is a meta-analysis confirming that adding protein to resistant exercise adds more benefit in terms of strength as well as more muscle mass. And I will finish here. Thank you very much. I'm available for questions. There is a question period, so do not hesitate. What is the best anti-aging factor we have around? Exercise. Yes.
I have a question for you.
Certainly.
What do you think about the relationship between osteopenia and sarcopenia? Are they completely separate processes? Or are they linked with one entirely?
No, the bone as well as muscle do have the same factors in terms of stimulants as well as